Once again, welcome to Science Circle. Today we will be discussing about an interesting topic, that is cell, which is the fundamental unit of life. The cell occupies the same central position in biology as the atom in the physical science. You can see the picture of different variety of cell. Slowly we will be discussing about the topics. So, all living organisms in the universe are made up of cells. They can exist as a single cell known as unicellular or a combination of multiple cells called as multicellular organism. Here are some examples of unicellular organism: amoeba, yeast, diatoms, Escherichia coli, etc. They can be prokaryotes or eukaryotes. Here are some characteristics of unicellular organism. Whereas multicellular organism is an organism composed of many cells. These organisms are developed by cellular specialization and division of labor. The organisms are generally visible in our naked eyes. They are eukaryotic in nature. Some of the common examples are like trees, humans, cats, and many more, which we generally see in our daily life. Let us study about the history of the cell. Cells are the basic structural and functional unit of life. They are the building blocks of life. Now, who coined the term cell? The term cell was coined in 1665 by the British scientist Robert Hooke. He was the first person to study cell under a microscope, and he examined a slice of cork under his microscope and he observed the honeycomb-like structures. The term initially derived from the Latin word cella, which means small room. Soon after his work, Antony von Leeuwenhoek made further discoveries by inventing his own microscope lenses that was powerful than the microscope of his time. He was the first person to observe human cell and bacteria under the microscope. With the help of electron microscope, the study of the objects smaller than cells became much more easier. We will be knowing about details of the microscope in further other videos. Now we will be seeing some interesting facts about cells. The single largest cell in the world is of ostrich. That is, the ostrich egg is the largest cell which we can see in our naked eyes only, but it is made up of only one single cell. The smallest cell are those of Mycoplasma gallisepticum, which is an organism intermediate between virus and bacteria, and it is very small in size. The largest human cell is the female ovum, which is 0.01 millimeter in diameter. The smallest human cell is the red blood cell. In humans, the number of cells is estimated to be about 1000 trillion. An average human cell measures between 0.01 mm to 0.03 mm. Nerve cell in animals are the longest cell. So you can understand the variety of cells which is found in all over the world. Now we will be discussing about cell theory. In 1838, Jacob Sheldon first proposed the idea that all plants consist of cells. In 1839, a little change came. Theodor Swan independently asserted that all animals as well as plants are made up of cells. Later, Rudolf Virchow presented the idea that all cells arise from the pre-existing cell. So, the cell theory comprises of the following postulates. Now you may be wondering, how do we study a cell, whether it is a plant cell or an animal cell? First, we will be observing an animal cell, that is the cheek cell. To observe the cheek cell, we need the following things, that is a toothpick, or ice cream spoon, slide with a cover slip, methylene blue stain or solution, blotting paper and microscope, that is the compound microscope. We have to scrape the inside surface of the cheek by using the toothpick or the ice cream spoon. Then we will transfer the material and spread it evenly on the clean glass slide. To cover this material, we will put a drop of methylene blue stain or solution in it. Stain is given to color the different parts of the cell so that when we will be observing under the microscope, we can distinguish the different parts of the cell. Finally, we will put a cover slip over the stained material. We should be very careful so that none of the air bubbles can enter while we are putting the cover slip. If there are air bubbles, it may hamper our observation. The excess stain which is there should be drained out by using the blotting paper. Now the slide is ready to be observed under the compound microscope. We will observe a spherical oval dot-like structure which is called nucleus near the center of each cheek cell, and a light transparent part which will be the cytoplasm, and the boundary wall will be the cell membrane. The different parts of the cell will be discussed in our upcoming videos. Similarly, we can observe plant cell under a compound microscope. For this experiment, we need a forceps, watch glasses, onion, it can be half cut or a full onion to be cut into 2 pieces, a needle, slide and cover slip, safranin, distilled water, dropper, blotting paper, brush and the compound microscope. To start this experiment, we have to follow some procedure. Firstly, we have to take a medium size onion and cut it into 4 to 5 pieces. Then we will remove the thin epidermal layer from the inner surface of the fleshy leaf of the onion. Then we will put it in the watch glass containing water to prevent the peel from getting dry or folded. Now we will take the pieces, if it is bigger in size, to make it small with the help of the scissors, because we don't need big pieces, rather than that we need small pieces. We will pass it to the next watch glass containing safranin stain and keep it for a couple of minutes. Finally, the extra stain of the peel is rinsed by dipping it in the watch glass containing water. Then we will transfer the specimen at the clean glass slide and put it in the center. We should be very careful that any part of the specimen is not rolled. If it is rolled, then with the help of the brush, we will make it straight enough. To mount the onion peel, a drop of glycerin is to be added on the slide before putting the cover slip. Glycerin is allowed to keep the specimen moist. While putting the cover slip, we should be very careful that no air bubbles enter, or else the observation of the specimen won't be correct. It won't be clear. Extra glycerin has to be removed by blotting paper. Now it is ready to be observed under microscope. Here we will be observing distinct cells which are rectangular in shape and have a distinct nucleus in it, which is darkly stained. Intercellular space is absent. Here cell membrane and vacuole is also visible, which remains unstained. On the basis of the structure of the nucleus, we divide cells into 2 categories, that is prokaryotic and eukaryotic cell, which we have already said about it in unicellular and multicellular organism. So in my upcoming video, we will be discussing in details about the eukaryotic and prokaryotic organisms. Thank you for watching my video. Please don't forget to subscribe my channel before closing it. Hope you will like my video. And please do share my video if you like it. Thank you.